This is Professor Capron. Welcome to this presentation on the impact of genetics, genomics, and epigenetics on growth and development. By the end of this presentation, you will be able to identify the impact of genetics on growth and development, explain the significance of genomics and the Human Genome Project, discuss the role of epigenetics in growth and development, and compare and contrast genetics, genomics, and epigenomics. Please pause the video and take some time to make sure you understand these key terms and their definitions. First, let's talk about gene pairs. Every human has 23 pairs of chromosomes for a total of 46 chromosomes. These chromosomes are structures within our cells that carry genetic information. We inherit one set of 23 chromosomes from our mother and another set of 23 from our father. These chromosomes are made up of DNA, which contains genes. Genes are the basic units of heredity and contain the instructions for making proteins, which are crucial for the structure, function, and regulation of the body's tissues and organs. For instance, genes determine whether a cell will become a muscle cell, a nerve cell, or a skin cell, among other types. This process is known as cellular differentiation. The genetic information within these genes not only dictates what type of cell it will become, but also influences how the cell grows and functions. For example, specific genes are responsible for producing growth factors, which are proteins that stimulate cell growth. Other genes might produce enzymes that help cells carry out their functions. Now let's explore the transmission of genetics, which refers to how genetic information is passed from parents to their offspring. Each child inherits half of their genetic material from their mother and half from their father. This means that traits such as eye color, hair color, and certain genetic disorders are determined by the combination of genes inherited from both parents. For example, if a child inherits the gene for brown eyes from one parent and the gene for blue eyes from the other, the child will typically have brown eyes because the brown eye gene is dominant. The genes we inherit influence many physical characteristics, such as height, skin color, and susceptibility to certain diseases. Additionally, some genes can also impact behavioral characteristics. For example, research has shown that certain genetic variations are associated with behaviors like risk-taking or aggression. However, it's important to note that behavior is influenced by a complex interplay of genetics and environment. So, genetic inheritance and chromosomes play a crucial role in determining how organisms develop and function. This genetic information influences a wide range of physical and behavioral traits. Heredity is the process by which genetic traits are passed from one generation to the next. It's all about the transfer of genetic material from parents to their children. For example, as we discussed, if a parent has blue eyes, there's a chance that their child might also have blue eyes due to the genes inherited from them. Think of heredity as the mechanism that ensures that traits like eye color, blood type, and even certain genetic disorders are inherited. This transmission happens through the genes contained in chromosomes, which are inherited from both parents. Each parent contributes half of their chromosomes to their child, so the combination of these genetic materials will determine the child's traits. Heredity focuses on how specific traits are passed down through generations and the genetic variation that arises from this process. Genetic variation is why siblings from the same parents may have different eye colors or personalities. It's also why genetic disorders appear more frequently in some families than others. Genetics is the scientific study of genes, genetic variation, and the way that these influence the traits and health of living organisms. It's not just about passing traits from parents to children, but also about understanding how genes work, how they vary among individuals, and how these variations affect biological processes. For example, genetics covers a wide range of topics, including gene expression, which is how genes are turned on or off in different cells and at different times, which affects how traits are expressed. Genetic mutations are changes in the DNA sequence that can lead to new traits or genetic disorders. Genetic counseling helps families to understand the risks of genetic disorders and the chances of inheriting or passing on specific traits. While heredity is specifically about the inheritance of traits, genetics covers both inherited traits and non-inherited factors that can affect health and development. For example, again, those inherited factors or traits pass from parents to offspring like genetic diseases. But non-inherited factors, that's where genetic research also explores environmental influences on gene expression, such as how diet or exposure to toxins might affect health or development. So, just to sum it up, heredity is a subset of genetics focused on transmission of traits from parents to offspring, emphasizing inheritance and genetic variation, while genetics, as a broader field, encompasses the study of all aspects of genes, including both inherited traits and how external factors can influence genetic information. The Human Genome Project, often abbreviated as HGP, is one of the most significant scientific endeavors of our time. The HGP was officially completed in 2003 after 13 years of intensive research and collaboration among scientists worldwide. This international effort was coordinated by the National Institutes of Health, the NIH, and the Department of Energy, the DOE, in the United States, along with numerous universities and research institutions across the globe. The primary goal of the HGP was to sequence the entire human genome, which consists of approximately 3 billion DNA base pairs. Sequencing the genome means determining the precise order of these bases, which are adenine, thymine, cytosine, and guanine, throughout the entire set of human chromosomes. This monumental task provided a comprehensive map of the genetic blueprint for building a human being. The HGP had several key goals, each contributing to its overall success and its impact on science and medicine. One of the core achievements of the HGP was determining the sequence of the DNA bases. This accomplishment means that scientists now know the exact order of the 3 billion base pairs that make up the human genome. This sequence information is crucial for identifying the locations of genes and understanding their functions. Another significant goal was mapping all of the genes in the human genome. It is estimated that the human genome contains about 20,000 to 25,000 genes. These genes are segments of DNA that carry instructions for making proteins, which are essential for the structure and function of the body's cells. The HGP has paved the way for significant advancements in genetic research and the field of personalized medicine. With the complete human genome sequence available, researchers can study genetic variations and their association with diseases. This research helps to identify risk factors for genetic conditions and developing new treatments. For example, identifying genetic mutations associated with certain cancers can lead to the development of targeted therapies that specifically address those mutations. One of the most promising applications of the HGP is in personalized medicine. This approach involves tailoring medical treatment to the individual characteristics of each patient based on their genetic profile. For example, pharmacogenomics is a field that studies how a person's genetic makeup affects their unique response to drugs. This knowledge allows for the selection of medications and dosages that are the most effective and have the fewest side effects for each individual. So, the completion of the Human Genome Project marked a significant milestone in science and medicine. Genomics is a field of biology that focuses on the comprehensive study of the entire genome of an organism. A genome is the complete set of DNA, including all of its genes. Genomics aims to understand the structure, function, evolution, and mapping of genomes. Genomics involves analyzing the entire DNA sequence of an organism to identify all the genes it contains and understand how they are organized and function together. This holistic approach provides insights into the genetic blueprint that guides the development, functioning, and overall biology of the organism. Genomics also examines how genes interact with each other and how these interactions influence the organism's traits and behaviors. Additionally, it looks at how environmental factors can affect gene expression and function. For example, how diet, stress, and exposure to toxins can influence genetic activity and contribute to health or disease. Genomics has several significant applications in medicine and research that have transformed our understanding of diseases and the development of targeted therapies. One of the primary applications of genomics is in disease research and diagnosis. By studying the genome, researchers can identify genetic mutations and variations that are associated with specific diseases. For example, certain mutations in the BRCA1 and BRCA2 genes are linked to an increased risk of breast and ovarian cancer. Genomic information can lead to more accurate diagnoses and the development of genetic tests to identify individuals at risk. Genomics plays a crucial role in the development of personalized medicine, which tailors medical treatment to the individual characteristics of each patient. By analyzing a patient's genetic information, healthcare providers can determine the most effective treatments and medications for that individual. For example, pharmacogenomics studies how a person's genetic makeup affects their response to drugs, as we discussed before. Genomics helps in understanding genetic predispositions. By identifying the genetic variants that increase the risk of diseases. Individuals can be informed about their unique susceptibility to certain health conditions. This knowledge allows for proactive measures to be taken to prevent or mitigate the impact of these conditions. For example, individuals with a genetic predisposition to heart disease can adopt lifestyle changes and receive regular screenings to manage that increased risk. Epigenetics is the study of heritable changes in gene expression that do not involve changes to the underlying DNA sequence. This means that while the DNA sequence itself remains unchanged, certain chemical modifications can affect how genes are turned on or off. These modifications can be passed from one generation to the next, influencing the traits and health of offspring without altering the genetic code itself. There are several key mechanisms through which epigenetic changes occur. DNA methylation involves the addition of a methyl group, which is CH3, to the DNA molecule, typically at cytosine bases. This modification can change the activity of a DNA segment without changing its sequence. When methyl groups are added to DNA, they can suppress gene expression, effectively turning genes off. For example, if a gene that suppresses tumor growth is methylated, it might be turned off, and that could increase the patient's risk of cancer. Histones are proteins around which DNA is wound to form chromatin, which is a substance of chromosomes. Histone modification involves the addition or removal of chemical group in proteins, which can influence how tightly or loosely DNA is wound around them. When histones are acylated, for example, the DNA becomes more accessible for transcription, leading to increased gene expression. Conversely, deacetylation makes DNA less accessible and reduces gene expression. Non-coding RNAs, or ncRNAs, are RNA molecules that are not translated into proteins but can regulate gene expression at various levels, including chromatin remodeling, transcription, and post-transcriptional processing. MicroRNAs, or miRNAs, and long non-coding RNAs, or LNCRNAs, are examples of ncRNAs that can bind to messenger RNAs, or mRNAs, and either degrade them or inhibit their translation into proteins. Epigenetic mechanisms have significance on growth and development and are responsible for the dynamic regulation of gene expression in response to environmental factors. Epigenetic modifications play a crucial role in early development, influencing processes like cell differentiation and organ formation. For example, during embryonic development, specific genes need to be turned on or off at precise times for cells to differentiate into various tissue types. Epigenetic changes ensure that these gene expression patterns are properly regulated. Environmental factors such as diet, stress, exposure to toxins, and physical activity can influence epigenetic modifications. For instance, a diet high in protein or certain nutrients like folate or B vitamins can promote DNA methylation, affecting gene expression patterns. Chronic stress can lead to changes in histone modifications, altering the genes involved in stress responses and potentially contributing to mental health disorders. So epigenetics involves studying heritable changes in gene expression that occur without any alterations to the underlying DNA sequence. Let's briefly compare genetics, genomics, and epigenomics. Genetics is the branch of biology that focuses on individual genes and their roles in inheritance. It involves studying how traits and characteristics are passed from parents to offspring through genes. Genes, which are segments of DNA, carry the instructions for making proteins, which are essential for various biological functions. Genetics looks at specific genes and their mutations or variations, and it helps to understand inherited disorders and how genetic traits are transmitted through generations. For example, cystic fibrosis is a genetic disorder caused by mutations in the CFTR gene. Genomics is a broader field that examines the complete set of genes or the genome of an organism. It looks at how all the genes interact with each other and with the environment to influence the organism's development, function, and health. Genomics involves mapping, sequencing, and analyzing the entire genome. It considers the interactions between multiple genes and their collective impact on the organism. The Human Genome Project is a notable example of a genomic study that mapped the entire human genome. Epigenomics focuses on the study of the complete set of epigenetic modifications that occur on the genetic material of a cell. These modifications can alter gene expression without changing the underlying DNA sequence. Epigenetic modifications include DNA methylation, histone modification, and non-coding RNA molecules. These changes can be influenced by environmental factors such as diet, stress, and toxins, and epigenetic modifications can be heritable and influence the development and health of the offspring. Each of these fields, genetics, genomics, and epigenomics, contributes uniquely to our understanding of developmental processes. Genetics identifies specific genes that are responsible for inherited traits and disorders and helps in diagnosing and treating genetic diseases at the individual gene level. Genomics provides a comprehensive view of all genetic interactions and their impact on the organism and enhances our understanding of complex traits and multifactorial diseases. Epigenomics explores how gene expression is regulated by epigenetic modifications and highlights the role of environmental influences on gene activity and development. The interplay between genetic, genomic, and epigenetic factors is crucial for understanding the full picture of growth and development. Genetic mutations can influence the risk of certain diseases, while genomic studies reveal the broader context of these risks. And epigenetic modifications can modulate gene expression based on environmental factors, adding another layer of complexity. Understanding this interplay helps in developing personalized medicine approaches that consider genetic predispositions, genomic interactions, and epigenetic influence. Now let's look at how we can apply this information to your practice of nursing. Genetic counseling is a crucial service that helps individuals and families understand their genetic risks and conditions. This involves assessing family history and genetic information to determine the likelihood of inherited conditions. Genetic counselors provide information on how genetic conditions are passed down through families and what the potential health implications might be. Genetic counseling helps to identify the risk of inherited diseases like cystic fibrosis, Huntington's disease, and certain types of cancer. It involves discussing the probability of passing genetic disorders to offspring and understanding the implications of genetic test results. Genetic counselors also play a critical role in advising patients based on their genetic information. This advice includes discussing the benefits and limitations of genetic testing, interpreting test results, and providing guidance on available medical options and interventions. Counselors offer support in making decisions about genetic testing and understanding the results. They help patients to understand the impact of their genetic makeup on their health and the health of their family members. Personalized medicine involves creating tailored treatment plans that take into account an individual's genetic and genomic information. This approach ensures that treatments are specifically designed to be most effective for the patient's unique genetic profile. Treatments can be customized for conditions like cancer, where targeted therapies are based on specific genetic mutations within the tumor. Preventive care in the context of genetics involves identifying individuals who have a genetic predisposition to certain diseases and providing early interventions to prevent or reduce the impact of these conditions. Early identification of genetic predispositions such as BRCA1 and BRCA2 mutations for breast and ovarian cancer allows for proactive measures like increased screening or preventive surgeries. Lifestyle modifications and preventive strategies can be implemented for conditions like heart disease or diabetes based on genetic risk factors. So, these are some practical applications in nursing that incorporate genetics, genomics, and epigenomics that have transformative potential in healthcare. Throughout this presentation, we've explored the profound impact that genetics, genomics, and epigenetics have on growth and development. Each field provides unique insights into how our genetic makeup and environmental factors influence our physical, cognitive, and behavioral traits. Remember that genetics focuses on individual genes and their roles in inheritance. It helps us understand the hereditary transmission of traits and genetic disorders. Genomics looks at the complete set of genes and their interactions within the genome, providing a broader view of how complex traits and diseases develop through gene interactions. And epigenetics studies heritable changes in gene expression that do not involve changes to the DNA sequence. It highlights how environmental factors can alter gene activity, impacting growth and development. The Human Genome Project, or HGP, was a landmark achievement that significantly advanced our understanding of genetics and genomics. Completed in 2003, the HGP mapped the entire human genome, providing a comprehensive reference for studying human biology and disease. It determined the sequence of DNA bases, which are the building blocks of our genetic material. It mapped all human genes, enabling researchers to identify genetic variations associated with diseases. And it has facilitated advancements in genetic research, personalized medicine, and our understanding of genetic predispositions to diseases. We looked at the distinctions and interconnections between genetics, genomics, and epigenomics, as it is crucial for comprehensively addressing growth and development. This presentation has highlighted all of these critical roles. Understanding these concepts is vital for anyone involved in healthcare, as it informs better diagnosis, treatment, and prevention strategies tailored to individual genetic profiles. It also emphasizes the importance of considering both genetic predispositions and environmental influences in promoting overall health and well being.